So we'll begin the week 7 problem solving session for the course NPTEL Experimental Biotechnology. So this week we'll predominantly look at questions related to immunology. We'll start with this basic question. Dash confers the immunity against cancer. And four options are given here: humoral mediated immunity, cell mediated immunity, innate immunity, and complements. So complements is complement system. So you can take a couple of minutes to think about this question, and whoever knows the answer can raise your hand. Okay, even if you don't know the right answer, you can share what you know about humoral mediated immunity, cell mediated immunity, innate immunity, and the complement system.
So the answer is cell-mediated immunity.
Yeah, but before that, can you do you know about other options?
Okay. Yeah, humoral mediated immunity is for it produce B cells produces the anti-plasma cells, differentiate into the plasma cells, and plasma cells produce antibody. It is for extracellular antigens.
Okay, okay. And innate immunity and complement system.
Innate immunity is by from the birth from birth. there is immunity like skin and mucus, etc.
Tears, saliva, and so on. Yeah.
Ah, yes, sir.
And complement system?
Like complement system, complements I have no much idea about it. I think opsonization, that category, I think so, sir.
Sorry, opsonization and what else?
I know only that opsonization, that's it, sir. I have no much idea about it.
Okay, so in opsonization, what is happening? Is it like you have coating of something, an antigen is being coated with something so that your uh, cells, cell mediated immunity can act? Is it something of that sort that you're trying to say?
For complement, sir.
Yes, yes.
Ah, yes, sir.
Opsonization is a process where you coat it with markers, right?
Oh,
An antigen is
yes, sir.
coated with markers so that your cells can recruit, I mean, identify them. Yeah, that's a brief overview. Good. But why do you think only cell-mediated immunity can confer immunity against cancer? Is there any particular reason?
So cell-mediated immunity involves T cells, like it activates T helper cells and T cytotoxic cells, which act against the cancer and virus, etc.
Okay. Okay. Good. Right. I'll first go through these options and then we also have a small flow chart of how this might possibly happen. So, other than your cytotoxic T cells, you also have these natural killer cells. And like you mentioned, these are involved in killing the tumor cells or virus infected cells. And like you had also mentioned, B cells turn into plasma cells, they mature, which produces antibody. And these are for pathogens that are outside the body, extracellular, and not cancer cells. Whereas innate immunity is the first line of defense. That's how you have these skin, saliva, tears that would be non-specific, and even your nostrils, the hair that you have there, all of these are a part of innate immunity. Yeah. Now, with respect to complement system, these will help the antibodies to lyse the pathogen, but cannot happen in anti-cancer defense. That's what they say. So, with respect to that, if we look at this, this is just a cycle of how cancer cells can be killed. So, we have something called as antigen presenting cells. So, once cancer cells can are exposed to these antigen presenting cells, these antigens are then um, collected and they are um, used for T cell activation. And these T cells are then recruited to the tumors, and these enter the tumors, and the cancer cells are recognized, and then they are killed. So specifically, this is a T cell mediated process. What we see here is where each of the process happen. For example, this activation of T cell happens in lymph nodes, and movement of T cells from lymph node to the site of tumor happens through blood vessels. Fine. Good. The next question is about antibody. Each antibody is made up of dash, two heavy chains and two light chains. Option B, two heavy chains and one light chain. Option C, one light chain and one heavy chain. Option D, one light chain and three heavy chains. So to start with, because this is a simple question, um, if anyone can share whatever they know about antibodies. Earlier we discussed that your B cells mature into plasma cells and then produce antibodies. Now, if you know anything else, you can still share, like recombination and other processes that makes antibody specific. You can share whatever you know. We'll then get onto the structure. And when anyone is trying to mention the structure, uh, you can also comment on if you know anything about variable chain or constant chain. and other structural aspects of the antibody. You can take a couple of minutes to think about it. Yes. Does anyone want to comment on this? The structure of antibody? Okay, even if you don't know the exact structure of the composition, can anyone tell me what is the maximum number of chains an antibody can have? Is it 3 or 4 or 2? You might have studied it as a Y-shaped structure.
Sir, it's having the Y-shaped structure, two heavy chain and two light chain.
Then do you want to comment on something else? Like other than having two heavy chain and two light chains?
Sir, one at the one end it is C terminal constant, and yes, sir. The upper portion is for the binding of the antigens, and there is a link between two heavy chains, that is disulfide. If I am
You have constant regions and you have variable regions. You have light chain and heavy chain, and these are linked to each other by disulfide bonds. And as you see here, these are the antigen binding sites, two sites. Okay. The next question is dash read region of the antibody is susceptible to protease cleavage? Option A variable, option B hinge, option C effector, and option D constant. Now, to think about this, you need to think about structure of proteins. So proteins have domains that are structured regions. So these structured regions, owing to the presence of hydrogen bonds, are not delicate, rather they are very constrained. So these are not very easy to cleave. But if there is any region that is not structured, for example, in proteins we have coils and loops and linkers that will connect different secondary structures within the same chain. So you can, for this particular question, you can think of, if there is any particular region that connects multiple structured domains. You can think about it that way. So here directly we can exclude the options variable and constant, because these are portions of the chain itself that are very large. So that leaves us with hinge and effector. Okay, so I hope you're able to see this. So what I wanted to show here was how the different chains are connected. So what you see here, if I hope you can see my pointer here, this region is the connector. These are all structured. You have, in the heavy chain, you have a variable region and a constant region, and in the FC region, which is the crystallizable region, you have two constant regions. Now these constant regions are all structured. This variable and this constant is also structured, but the link between them is unstructured, which is what would cleave them. There is also an enzyme that people specifically use to cleave this. Is anyone aware of it? That would basically cleave your these constant chains.
So pepsin and papain.
Sorry, pepsin and
Papain.
papain. Yeah, good. Yeah, that is a very well known enzyme that is known to cleave these hinges. Good. The next question is, dash is not an antigen presenting cell, and four options are given here: macrophages, B cell, T cell, and finally dendritic cell. So before we go to the right option, you can think for a couple of minutes and then first answer what antigen presenting cells are. Then we'll take it from there. What is the function of antigen? I mean, it's there in the name, but you can still think and tell what an antigen presenting cell is there and why there is a requirement for an antigen presenting cell. You can take a couple of minutes to think about this. So does anyone want to try answering what an antigen presenting cell is?
Sir, may I?
Yeah, 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 sure.
So antigen presenting cells are nothing but whenever macrophage T cell or dendritic cell encounters an antigen,
Yes.
it binds to it, and then it's going to process it. Means it going to break down into fragments. Then it will display on the membrane through MHC class 2,
Hmm.
and then the T helper cells identify that antigen fragments on the MHC class 2, and the TH gets activated, and this will activate the other B cells and T lymphocytes, and antibody gets released.
Okay, but why do antigens need to be presented? Is there any particular reason why antigens need to be presented, and why can't?
For specificity,
Sorry, yeah. What is the necessity for antigen presentation?
specific antigen, like antibodies need to bind to the specific antigen, so they're going to process it.
Can't your plasma cells or B cells directly take up your antigen and then take care of it? Is that possible?
Like what, sir? Can you repeat?
Yeah, yeah. So what I'm asking is, at the end, it's your B cell or your T cell that is involved in, uh, let's say, eliminating your pathogen. So
Yes, sir.
can't these directly get involved in identifying or interacting with the antigen? Do you need a cell that would actually function as an antigen presenting cell? Why would that be the case? Can you just think about it? Did you get my question?
Yes, sir. I got you.
Yeah, yeah.
I was thinking.
Okay. You can think about it in terms of location. You can think whether B cells and T cells roam around your body in the blood vessel, and how probable it is for them to encounter antigens. And
So
if
B cells are present at lymph and blood. In liquid, it is present, sir. B lymphocytes.
yes, but the ones in the blood, are they capable of accepting antigens?
Um, no, I
Yes,
think so, sir.
yes. So
Directly, no.
yeah, so the reason you need an antigen presenting cell in the first case is because these are the cells that are predominantly present in your blood vessels. I mean, they can come into contact with anything that is foreign to the body. And whereas your B cells and T cells, they are specialized. So that's why you have these classification. I hope you got that. Okay. So now, which of these are antigen presenting cells? Macrophages, B cell, T cell, and dendritic cell. Which of these are actually antigen presenting cells?
A macrophage and dendritic cell.
Okay. And which of these are not?
I, T cell is not an antigen presenting cell.
Okay. I'll go to the slide. Give me a minute. Okay. So yeah, like you had mentioned, macrophages, dendritic cells, and B cells. So they can process the antigen and present on MHC molecules to the T cells to start your adaptive immunity. While your T cells can respond to antigens, but they cannot act as antigen presenting cells themselves. And even in this, you have a further classification. Dendritic cells are called as the professional APCs, and along with it, you have macrophages. Um, your neutrophils and natural killer cells, they can interact, but they cannot present antigen. Right. The next question is, dash cells differentiate to form the plasma cells? macrophages, B cell, T cell, dendritic cell. So we discussed this as a part of another question. So if you can remember, you can let me know. We talked about
B
how,
cells.
yes, so those are the regions where you have antibody. And this is just a schematic. What you see here is T cell dependent activation and T cell independent activation for your B cell to be able to form antibodies. So in your T cell dependent activation, you get this MHC class 2 that interacts with your T cell, and one of these two things happens. Either you get your memory B cells, I mean you get a fraction of both. You get your memory B cells, and you also have these plasma B cells. These produce your antibody. These respond when you have a similar antigen later in your life. This acts currently. In the T cell independent activation, you have these pink blobs are your antigens. Again, these would interact with these memory B cells to be able to produce your antibodies. IgM is immunoglobulin M, which is a predominant class of antibody. Yes. So that's how B cells can differentiate to form plasma cells. Yes, this is just a textual version of the same. So the activated B cells, like we saw in the figure, it can either secrete antibody or provide long-term immunity. So depending on how they mature into plasma cells or B cells, it changes. Fine. The next question is, monoclonal antibody is generated by dash hybridoma technique, purification of antibodies from the serum, SDS page, and Western blotting. So before we go to the right answer, you can think for a couple of minutes and tell what is monoclonal, what would monoclonal represent before we go on to the method for the antibody generation. So when someone says monoclonal antibody, what do they mean? You can start with that, and then we'll get back to the procedure to be able to generate these monoclonal antibodies.
Sir, monoclonal antibodies are produced from one specific epitope of on antigen. Like it is very specific towards only one epitope on the antigen.
Right. Now that you mentioned about epitope,
Yes,
uh,
sir.
yeah, what is an epitope?
So an antigen consists of epitopes. With the help of epitope, it is a binding site to the antibody.
Okay. Can one epitope be, can more than one antibody bind to one epitope?
Uh, if it is specific, no, sir.
Yes, yes. So,
It may be, it can bind, sir, actually.
but can one
Yes, sir.
Can you think about it again? We are talking about specificities here. So the question was, can one antibody bind to multiple epitopes, and can one epitope bind to multiple antibodies? Only one of these statements is true.
Second option is correct, sir. Like anti. Uh,
Yeah. So in your antigen, you will have multiple regions. Depending on which region is being presented, which, let's say we are talking about a particular protein on a pathogen, a particular segment of the protein could alone be presented to your B cells to be able to form your antibodies. So depending on that region, you will have these antibodies interacting. So, and within that region, there could be some that are exposed uh, to a higher proportion and some that are exposed to a lower proportion. This means that you can have more than one antibody that can target the same epitope, but one antibody can bind to only one epitope, because uh, here is where your monoclonal concept comes in, which means that the smallest region of epitope that is being recognized and the plasma cell produces the antibody specifically to that, that is called monoclonal, as we see from the figure here. This is called as this, what you see here, a very small fragment that is being presented, and that creates the formation of antibodies. Right, good that you had mentioned about epitopes here. And so coming back to the techniques here, when we purify antibodies from the serum, why will it not be monoclonal? Does anyone have an answer for that?
sir, can you repeat the question?
Yeah, I'm talking about this option B. When we purify antibodies from the serum, why will that not have, why will that not be monoclonal? Or will it be monoclonal? Okay, what do we mean by serum here? We'll start from there. Do you people know what serum is?
So blood minus plasma is serum.
Yes. So you take your blood, you centrifuge it, remove the cells and the liquid that you have. Once your cells are precipitated, the liquid that you have, that would be your serum. So what all could this contain?
Sir, in serum there is no clotting factor.
Yes, we have. Okay, clotting factor is not there. What else? Are there any cells in serum? Do we have any cells in serum?
The B cells,
Okay.
I B lymphocytes, I think so.
Okay. So now, will it have monoclonal antibodies, or now that you've told about B lymphocytes, will it have monoclonal or uh, polyclonal antibodies?
It depends upon the antigen, I think so, sir.
No, no. So here we're talking about humans, right? So that means that you will have exposure to multiple antigens that are so within the body you will not have only one antibody. Do we agree on that?
Yes, sir.
Yes. So in that case, in your serum, will you have only one antibody?
No, there will be multiple antibodies.
Yes. Good. So that's the reason we cannot identify, or extract monoclonal antibodies from serum, because that will have antibodies towards multiple pathogens and multiple antigens, not pathogens, multiple antigens. Which is why we use this technique called hybridoma technique. And here we take a mice, immunize it with the antigen. This will help us form these mature B cells. Now we fuse these B cells with myeloma cells. So myeloma are carcinogenic. Now the point of using this carcinogenic cells to B cells, which are mature and can form antibodies, is that this will act as a bioreactor that will produce antibodies. So these will produce antibodies infinitely, and you have selections involved that would also screen your hybridoma for the antibody that you are interested in. So in this process, since we have specific B cells that are isolated against an antigen, it can produce antibodies specific to that one epitope alone. That is why this method exists. So, in short, this hybridoma technique can help us in generating monoclonal antibody. Right. A continuation question. So, during monoclonal antibody production, B cells are isolated from dash, and we have four options: blood, spleen, liver, and lymph. You can take a couple of minutes to think about this question. Okay, does anyone know where these B cells that are used for monoclonal antibody production, like we mentioned here, are isolated from? cells are derived from spleen, and then they are fused with myeloma, and we have discussed this. So we'll come to the wrong options. So blood. Um, Two reasons: it could contain other B cells as well. So we'll not be able to specifically isolate the for the one that we are interested in. In liver, your B cell does not mature. Lymph node. Here, although it contains B cells, we still use spleen.
Sir, why not the lymph node? Oh, there also contain B cells.
I think it could also be due to um, antigen presentation and maturation, because merely containing B cells might not give us an antibody producing B cell. Where exactly does B cell mature?
And the bone marrow.
Okay, and where is your anything specific within the bone marrow? Is it the lymph node or spleen?
And it's lymph node.
Maturation happens in the lymph node.
Oh,
Is it? Okay. Then I need to
yes.
check that. I was under the impression that this would happen in the spleen. I can check and I'll see that. I was sure that it doesn't happen in your blood or liver, but I'll check that. Thanks for
Okay, sir.
thanks
Thank you.
for pointing that out. Okay, I need to check this. Fine. The next question is: antibody titers can be detected by, and four different methods are given here: ELISA, microscopy, northern blotting, and HPLC. Yeah. So for this question, we had discussed earlier as well. Does anyone want to try expanding what ELISA is?
So it is enzyme-linked immunosorbent assay.
Okay. So why would other methods like northern blotting, microscopy, and HPLC not work? Do you have any idea?
So northern blotting is for DNA, not for proteins.
Okay.
Sorry, it's RNA or DNA. I don't remember.
Okay, it's for nucleic acids.
Yeah, nucleic acids.
And HPLC? HPLC is for small molecules or small peptides and small nucleic acids. It cannot, the columns that people use for HPLC, which are predominantly C8 or C18 columns, cannot accommodate antibody. And microscopy, it is very less defined in this option. Um, Although you can do quantitative studies with microscopy, still antibody titers cannot be uh, detected unless you have it tagged with a fluorescent marker, which normally doesn't happen. And so ELISA is a method because you use antibodies that are specific to your target of interest, and you also have an enzyme like a horseradish peroxidase that would illuminate if you give a particular substrate. And the amount of horseradish peroxidase itself is dependent on how much units of the antibody is bound. So this is the method, standard method of choice, gold standard that people use for detecting um, or obtaining the antibody titers. The next question is, dash is the process of linking together of antigens by antibodies and formation of visible aggregates. Four options are given here: agglutination, precipitation, proliferation, and gelation. So, if you have ever undergone immunology practicals during your college coursework, you might have done this one particular assay where you take a gel, you add antigen to it, and you add antibody to it, and you see it diffusing and forming a pattern. The reason you could see this pattern is because it forms this aggregate. Now the question is, what is this process of formation of this aggregate called as? So think about this for a couple of minutes. If you are not very sure, you can start with eliminating the wrong options, like we use, like we usually do.
Sir, answer is agglutination.
Okay, why is it not precipitation?
So precipitation is the formation of insoluble complex when soluble antigens react with antibodies.
Okay, and
But
in agglutination?
agglutination, binding of antigen to the antibody, but it is not soluble like precipitation.
So why are we able to see it? Why is it visible? Any idea? If it is,
Like visible clumps, sir. I mean, sir, what you're trying to ask.
yes.
It is due to binding, I think so, sir. I don't have much idea. Due to that, which forms clumps. It is insoluble. That's the reason we can observe it. I think so.
Okay. Yeah, right. What you mentioned is right. And I'll just go through the wrong options once. Proliferation, it's nothing but growth or um, making of new cells or achieving different growth stages that we know. Gelation, this has nothing to do with the immunology that we are discussing here. It's just changing of phases from solid to liquid or an intermediary liquid to solid. So it's a phase change process. Precipitation is a term that people use commonly with respect to highly insoluble complexes. Now, there is a difference between aggregation and uh, precipitation. In precipitation, we majorly talk about these insoluble complexes, but these visible aggregates need not be insoluble. These can still be soluble, but still be visible. The reason that they are visible is because they are large and they are all focused and localized. Agglutination is specific in immunology, where antigens can be bound to antibodies. And one thing that wanted to mention here was that the way blood group is identified or determined is based on this agglutination, where you have uh, markers on your red blood cells that are used to directly identify, uh, based on this visible agglutination, whether you belong to a particular blood group type. So this brings us to the next question. In this blood group typing, is it indirect agglutination or direct agglutination? How do we identify the blood group? Now that we've known that blood group typing is based on agglutination and not precipitation and gelation, we can exclude the other options. But is the type of agglutination direct or indirect? Okay, to be able to differentiate direct and indirect agglutination, you can think of it as whether your antigen is directly bound to your antibody, or is your antigen also bound to some other carrier molecules. Like for example, in commercial kits, you have these bound to covalently linked to beads made out of agarose or latex. So if you have,
It's directly bound to the antigen.
yes, so in blood group typing, it is direct agglutination because you just take your blood, you have none of these carrier molecules, you have the antigens present on the RBCs that would help you identify your blood group. Right, this is just a schematic to tell you how agglutination happens, and this visible clump is a result of your agglutination. Fine. The next question is, which of these is an anticoagulant? EDTA, phenol red, KCl, and glucose. So you can think about, you can first, before you start answering this question, you can first let me know what coagulation is and whether it is a, or and then what are all the chemical species that are involved in coagulation. Is there an ion involved, heme involved, and so on. So we'll start with what coagulation is and then we'll take it from there.
It's like a clot formation, that liquid blood form in the clot solid state.
Okay, so when you have change of phase from liquid to solid, which happens in a blood clot formation, that is coagulation, right? Okay, so now for this clot formation, do you need any specific chemical species in the absence of which this clot will not happen?
Sir, can you please repeat the question?
I wanted to know if you So what is actually forming these clogs in coagulation? Is this an enzymatic process?
Yes, there might be some enzyme that help to clot the blood.
Okay, so are there any specific molecules like fibrin or any polymers or biomolecules that you think are involved in coagulation?
Fibrinogen.
Okay, and since you had mentioned that this is an enzymatic process, enzymes involve metal ions, and metal ions in this particular case that is of relevance is calcium. So calcium is involved for the enzymes to function, which is a cofactor. In the presence of EDTA, calcium will be chelated, meaning the enzymes will be stripped off of the calcium ions. Now, because the enzymes are stripped off of the calcium ions, they cannot function anymore. So, your enzymatic formation of clogs is prevented. So, here among these four, EDTA is the one that can strip off this calcium ion. Phenol red, that is a pH indicator. Glucose has nothing to do with any of these. So is KCl. Right. The next question is dash is an example of immunodiffusion test. And four options are given here: Otterloni method, ELISA, Western blotting, Northern blotting. Note that we are talking about immunodiffusion, not just immunological test.
Sir, it is Octaloni method.
Okay, so what happens in this method?
Sir, in this method, the antibody binds with the antigen and the agglutination takes place in the case.
Yes. And do we have any specific patterns? Are you able to see my screen? I am sharing three different patterns here.
Yes, sir.
Yes. So, based on what you see on the screen, are you able to recall what these patterns could mean? Or okay, if not that, you can tell me what you know about this method or this test, and then I'll take it from there.
Sir, in the case when the antigen concentration is equal to antibody concentration, so total amount of antigen and antibody is agglutination. In the case, the first structure is formed.
The top one, is it?
Yes, sir.
Okay. What about these?
Or in the other case, when the antigen is concentration is not equal to antibody, so this other structure is formed in that case.
Okay, just to give a clarity on this, we have one antibody, we have five antigens, and we have a control here. And now it is diffusing. Or like you can see from here, you can see my pointer here, I believe. So this pointer here, this is your antibody. The other two are antigen in all the cases. So what you told is the inverse. So this intersection and this crossover happens when you have non-identity, whereas when you have identity, you don't have this crossover. This means that the antigen and antibody are one to one. It has nothing to do with the concentration per se, but about if the antigen and antibody are specific to each other. Uh, why did you say that the first one is the one where you have this agglutination and visible line? Can you please repeat that?
Sir, I'm not sure about it.
Okay. So what you can remember is, if the reactions are specific, you will not have this interaction. When you have some non-specificity, that is when you have this diffusion that leads to, this intersection across antigen antibodies. Fine. The next question is, dash of the precipitin ring in radial immunodiffusion is directly proportional to the concentration of the antigen. Yeah, here you can apply your knowledge on the concentration and let me know how this is different from the previous one. Okay, first we look at what this method called radial immunodiffusion is. Here again your antigen diffuses into agar containing the specific antibody, and this interaction forms this precipitin ring. Now let's say we do a standard curve with this for specific concentrations of antigen. What is the radius of the precipitin complex? With that we'll know for what concentration of antigen what will be the ring diameter. With this we can plot a standard curve. Now when we have the standard curve, we can take any antigen, any volume, and know how much amount of it by just calculating the diameter. This is how, based on this, we get to calculate the concentration of the antigen. Right. The next question is, with the help of immunoprecipitation, we can understand dash of a protein. Yeah, this is a very simple question. In all the previous slides, we were discussing about antigen antibody interaction. So, this should tell us something about interaction. So, can anyone summarize what immunoprecipitation is? this is an application of immunological techniques to biochemistry or structural biology. So we know that antibodies can be raised against any targets. So what we do is we decide what protein we are interested in. We raise antibodies against that protein. We raise antibodies against that protein. Then within the cell lysate, we will add antibodies so that we can capture our protein of interest. Now this will be like a bait and a target. Now after that, we use a solid support that will bind to our antibody, and the protein of our interest will be bound to other interacting proteins. So with the help of this immunoprecipitation method, we can identify interacting partners for a particular protein. And the last question is, the reagent used to develop an HRP based secondary antibody system in Western blot is dash. Option A luminol H2O2, option B BCIP and BT, option C dimizone, and option D streptavidin. Can anyone tell me what HRP based secondary antibody system is? Because just expanding HRP would give us the right answer here. Can anyone try answering that? Okay, HRP is horse radish peroxidase. Now, this is an enzyme which will convert peroxide, and in the presence of this luminol reagent, we can get luminescence. Now, by covalently linking this to an antibody, we can visualize, like in the case of Western blot. So the right answer here is luminol H2O2. Fine. So with this, we come to an end of today's questions. If you have any questions, you can let me know. I'll be on the call.
So, what is the role of luminol in this HRP-based secondary antibody?
HRP is your horse radish peroxidase. You need a quantitative metric that will tell you whether this H2O2 has been acted upon by horseradish peroxidase. So the product of HRP acting on H2O2 will allow luminol to be excited. That is where we get our visual readout. So when HRP is acting on H2O2, you get some product that will allow luminol, and that the interaction between that product and luminol will give luminescence, which can be captured by a scintillator or a camera. Was that clear?
Yes, sir. Thank you.
Okay, thank you.
So it's the anticoagulant that is lymph node or spin, the lymphocyte formation.
Let me go to that question, read that question once again. Give me a minute. This question is what you're asking, right? Monoclonal antibody production. B cells are isolated from
Yes, sir.
lymph node or spleen. I'm not very sure about the answer. From what I see, it is mentioned that because spleen is vascularized, you will get more B cells from the spleen compared to the lymph node. But it doesn't say that in the lymph node you will not have B cells. So, what I assume is because you get more, because the yield is more, people prefer the spleen compared to the lymph node. That is my understanding.
Okay. Okay.
Yes. Any other
Thank you, sir.
questions?
No, sir. Thank you.